Welcome to the deep dive. Today, we're jumping into CRISPR. It's this gene editing technology that, well, it arrived with this mix of incredible hope and, frankly, some existential dread. And you can really see that duality in two big moments, just 5 years apart. First, late 2023, we get FDA approval for Casgevy. That's for sickle cell disease, a real cure, using somatic therapy. But then you rewind to November 2018, and that shadow. The news about He Jiankui in China, secretly editing human embryos, twin girls born with altered genes. That's heritable germline stuff.
Exactly. And that's the core of it, isn't it? We're at this fork in the road. One way is precision medicine fixing a specific genetic issue in one person. That's somatic therapy. The other path, that's about changing our actual evolution, making heritable changes passed down through generations. That's the germline issue. So our goal today is to kind of unpack that difference.
Okay, right. Let's break down the science a bit first. So
Yeah.
CRISPR, it wasn't invented in a lab, was it? It's older than that.
No, not at all. It's actually repurposed from bacteria. It's their ancient immune system, basically.
Yeah.
How they fight off viruses. We just sort of borrowed it, turned this natural defense into a tool.
Borrowed an evolutionary weapon. Okay, so how does it work? I hear this analogy about word processors.
Yeah, that's a pretty good way to think about it. You've got two main parts. There's the guide RNA. Think of that as the find function. It's like a homing beacon that takes the machinery right to the specific DNA sequence you want to target. And then there's the Cas9 enzyme. That's your molecular scissors. It makes a clean cut, a double strand break, right where the guide RNA tells it to.
And the replace part, the actual edit.
Well, that's the cell's own repair kit kicking in. Once the DNA is cut, the cell tries to fix it, and we can sort of nudge that repair process to make the change we want.
Got it. Okay, so let's talk about the success story then. Casgevy for sickle cell. That's where it gets really exciting, right? A single faulty gene causing the disease.
It really is fascinating. And the strategy they used is quite elegant, actually. It's indirect. Instead of trying to, directly fix the broken adult hemoglobin gene, which is tricky, they used CRISPR to switch off another gene entirely. It's called BCL11A. Normally, this gene acts like a switch that turns off fetal hemoglobin production shortly after birth.
Fetal hemoglobin, the stuff babies have.
Exactly. It works perfectly well, but we usually stop making it. By knocking out BCL11A, they basically jam that off switch. So the body just keeps producing the functional fetal hemoglobin, and that fetal hemoglobin compensates for the faulty adult kind. It effectively cures the disease.
Wow, so you're reactivating an older working system instead of fixing the newer broken one. That's clever.
It is, and critically, this is done ex vivo.
Meaning outside the body.
They take the patient's stem cells out, edit them in the lab where they can do quality control, check everything is okay, and then infuse the corrected cells back into the patient. That gives you a huge safety advantage.
Right, that control factor. Editing cells in a dish versus editing an entire developing embryo. Which brings us to the ethical line.
The germline red line, yes. Somatic edits, like the sickle cell treatment, affect only the patient. They aren't passed on. Germline edits made to sperm, eggs, or embryos are heritable. They change the genetic makeup of all future generations. That's the massive difference.
And that's what He Jiankui did, editing the CCR5 gene in embryos, claiming it was to make the twins resistant to HIV.
Yes, but the scientific community reacted so strongly because, well, first, the intervention just wasn't necessary. Medically gratuitous is the term often used. We already have very effective ways to prevent HIV transmission from parent to child, like sperm washing combined with antiretroviral therapy. The risk was already near zero.
So he exposed these children to completely unknown, permanent genetic risks for basically no good reason.
Pretty much. You can't ethically justify altering the human gene pool forever to solve a problem that you can already manage safely with existing methods. That was the core ethical breach, alongside the secrecy and lack of oversight.
Okay. Beyond the ethics of that specific case, why is the broader scientific community so against germline editing right now? You hear about safety concerns.
It really comes down to technical safety. The technology just isn't precise or reliable enough for something with such permanent consequences. There are two big worries.
Which are?
First, off-target effects. The CRISPR machinery might make unintended cuts elsewhere in the genome, places you didn't target.
Like typos far away from where you meant to edit.
Exactly. And these off-target cuts could hit vital genes, maybe even genes that suppress tumors, potentially causing cancer or other diseases down the line. We just can't predict or control that well enough yet in embryos.
Okay, off-target cuts. What's the second major risk?
That's mosaicism. When you edit a very early embryo, it's already dividing. The editing might not work perfectly in all the cells, so you end up with a mix, a mosaic of edited and unedited cells throughout the person's body. Some tissues might have the edit, others won't. Critically, this includes the germ cells, sperm, or eggs.
Meaning the edit might be passed on unpredictably, or not at all, or only partially. That sounds incredibly messy.
It is. It makes the outcome for the individual unpredictable, and the consequences for future generations even harder to foresee. It's a level of uncertainty that's just unacceptable right now for heritable changes.
So, zooming out, we've got this incredibly powerful tool, miracles on one hand like Casgevy, and this huge ethical danger zone with germline editing on the other. The technology seems to be racing ahead of our ability to manage it.
That's the crux of it. The pace is so fast, it forces us to confront a fundamental question.
Yeah.
It's not just about can we rewrite our own genetic code, but should we? Especially when it comes to changes that affect not just one person, but potentially everyone who comes after them. Are we ready as a global society to make eventually irreversible decisions about the human blueprint that needs careful, sustained, worldwide discussion, far more than we're having right now?